Hey guys, welcome to another episode of the Human AI podcast. This is part 3 of glycolysis, electron transport chain, glucose homeostasis, and glycogen metabolism. I'm here with Grog, who's going to help us understand this topic. Take everything that Grog says with a pinch of salt. I hope you find this informative. Let's continue, guys. Dear Grog, can you explain us again? So we have electrochemical energy generated in the intermembrane space. Okay, and we've talked. We have difference in charge and difference in proton concentration. I understand that. Okay. Let's go forward from that. Let's talk a bit about the energy yield from oxidation of one molecule of glucose. Shall we?
Haha, let's do this. Starting from the electron chemical energy in the mitochondrial intermembrane space, proton gradient from charge and concentration differences, let's dive into the energy yield from oxidizing one glucose molecule. One glucose, through complete aerobic metabolism, glycolysis citric acid cycle, electron transport chain, yields around 30 to 32 ATP. Breakdown: glycolysis, hydrocal compound. The number of ATP is 4 glucose. Cytosol gives 2 ATP and 2 NADH. 1,5 ATP via ETC using malate aspartate shuttle. Pyruvate oxidation, mitochondria, produces 2 NADH, around 5 ATP. The citric acid cycle, twice per glucose, yields 2 GTP, around 2 ATP, 6 NADH, minus 15 ATP, and 2 FADH2, minus 3 ATP. The ETC uses NADH and FADH2 electrons to pump protons, powering ATP synthase. Total, around 30 to 32 ATP, depending on shuttle efficiency and proton leaks. Clear? Want details on ATP synthase or NADH, FADH2 yields?
Okay, no, that's okay. I want to ask you about uncoupling reagents.
All right, let's talk uncoupling reagents, keeping it concise. Uncoupling reagents disrupt the proton gradient across the mitochondrial inner membrane, preventing ATP synthesis by ATP synthase. Normally, the electron transport chain, ETC, pumps protons into the intermembrane space, and their flow back through ATP synthase drives ATP production. Uncouplers, like 2,4-dinitrophenol, DNP, or therogenin in brown fat, act as proton shuttles, letting protons leak back into the matrix without passing through ATP synthase. This dissipates the gradient as heat, uncoupling electron transport from ATP synthesis. So energy from glucose oxidation is lost, not stored as ATP.
So protons go back to the matrix, you say?
Yeah, exactly. Uncoupling reagents let protons flow back into the mitochondrial matrix across the inner membrane, bypassing ATP synthase. Instead of using the proton gradient to make ATP, the energy is released as heat, breaking the link between electron transport and ATP production. Clear? Want to dive into
What's
example?
the point of this, though? And tell me about brown fat thermogenesis, too.
Uncoupling reagents and brown fat thermogenesis both bypass ATP synthesis to produce heat, but their purposes differ. Uncouplers like DNP disrupt the proton gradient in mitochondria, letting protons leak back into the matrix without ATP synthase, wasting glucose oxidation energy as heat. Historically, they were used for weight loss, burning fuel without storing ATP, but they're dangerous, causing overheating or organ damage. Brown fat thermogenesis, however, is a natural process in mammals, especially newborns or hibernators, to generate heat. Brown fat cells have thermogenin, UCP1, a protein that uncouples the proton gradient, allowing protons to return to the matrix, dissipating energy as heat instead of ATP. This helps maintain body temperature in cold environments. burning fatty acids and glucose for heat production. Clear? Want more on thermogenin's regulation or uncoupler risks?
I want to ask you about thermogenin and UCP1.
Thermogenin, also called UCP1, uncoupling protein 1, is a protein in the mitochondrial inner membrane of brown fat cells that drives thermogenesis. It uncouples the proton gradient by allowing protons from the intermembrane space to flow back into the matrix, bypassing ATP synthase. This dissipates the energy from glucose and fatty acid oxidation as heat instead of ATP, helping maintain body temperature, especially in newborns, hibernators, or during cold exposure. UCP1 is activated by fatty acids and regulated by norepinephrine, which signals fat breakdown and heat production. Clear?
So put everything into a nice story for me that we've talked about in these last bits, the last few messages.
All right, let's weave a story that ties together the electrochemical energy, glucose oxidation, uncoupling reagents, brown fat thermogenesis, and thermogenin UCP1, keeping it clear and concise. Picture a bustling city inside your cells, where glucose is the main fuel powering everything. This city's energy factory is the mitochondria, and its electron transport chain, ETC, is like a water wheel, spinning as electrons from glucose flow through, pumping protons out to the intermembrane space. This creates a proton reservoir up high, with a charge and concentration difference, an electrochemical dam ready to burst. Normally, protons rush back into the matrix through ATP synthase, a turbine that churns out ATP, the city's energy coins. One glucose molecule fuels this whole system, yielding about 30 to 32 ATP through glycolysis, the citric acid cycle, and the ETC, keeping the city humming. But sometimes, the city needs heat, not coins. Enter uncoupling reagents like DNP, sneaky saboteurs that poke holes in the dam, letting protons leak back into the matrix without hitting ATP synthase. The energy from glucose burns off as heat, like a fire without purpose, once used for quick weight loss, but risky, overheating the city. In contrast, brown fat cells are the city's cozy heaters, especially in babies or cold climates. They have thermogenin, or UCP1, a natural gatekeeper in their mitochondria. When it's chilly, norepinephrine signals UCP1 to open, letting protons flood back into the matrix, skipping ATP synthase. This turns glucose and fat into a warm glow, keeping the body toasty. So, while the ETC builds the dam to power ATP, uncouplers and UCP1 choose heat over storage, each with its own role in the city's energy tail.
This story was great, man. Thank you. Now I understand. Okay, let's talk a bit about mitochondrial DNA now. Why does it have a closed circular double-stranded molecule? And why does it code for respiratory chain proteins? How does the structure relate to the function? And
Glad you love the story. Let's dive into mitochondrial DNA, mtDNA, keeping it concise. Mitochondrial DNA is a closed, circular, double-stranded molecule, resembling bacterial DNA, because mitochondria likely evolved from ancient bacteria. This compact structure, about 16,500 base pairs in humans, fits neatly in the mitochondrial matrix and resists damage. It encodes 13 proteins, all crucial subunits of respiratory chain complexes, I, 3, 5, and ATP synthase, plus 22 tRNAs and 2 rRNAs for mitochondrial protein synthesis. Why respiratory chain proteins? These complexes drive ATP production via the electron transport chain, ETC, and mtDNA's local encoding ensures quick, efficient production of core components right where they're needed. The circular structure supports rapid replication and transcription without loose ends, matching the mitochondria's high energy demands. Nuclear DNA encodes most other mitochondrial proteins, but mtDNA's focus on ETC components ties its structure to its role as an energy hub. Clear?
Okay, let me ask you about maternal inheritance. So, the embryo derives the mitochondria from the egg, and most sperm mitochondria are not absorbed. So why? How come? Why is this? How is this possible?
Maternal inheritance of mitochondrial DNA, mtDNA, happens because the embryo gets its mitochondria almost exclusively from the egg. The egg contributes thousands of mitochondria, packed with mtDNA, to the embryo. Sperm carry a few mitochondria, about 100, but these are typically not absorbed or are actively degraded post-fertilization. In mammals, sperm mitochondria enter the egg but are marked with ubiquitin, targeting them for destruction by the egg's cellular machinery, like lysosomes. This ensures only maternal mtDNA persists. Why? It likely evolved to avoid mtDNA conflicts, ensure genetic consistency, and reduce transmission of sperm mtDNA mutations, given sperm's high metabolic stress. The egg's large mitochondrial pool also dominates functionally. Clear? Want more on the degradation process or evolutionary reasons?
No, that makes sense. Okay. So, oxidative phosphorylation declines with age. And this seems to be because we have a lot of mutations to the mitochondrial DNA.
You're spot on. Oxidative phosphorylation, the process making ATP via the electron transport chain and ATP synthase, declines with age, partly due to mitochondrial DNA, mtDNA mutations. mtDNA's proximity to the electron transport chain exposes it to reactive oxygen species, ROS, which cause mutations over time. Unlike nuclear DNA, mtDNA lacks robust repair mechanisms and protective histones, so damage accumulates. These mutations disrupt genes encoding respiratory chain proteins, complexes 1, 3, 5, ATP synthase, impairing ATP production. Aging tissues, like muscle and brain, show reduced energy output as mutated mtDNA multiply. Clear? Want more on ROS damage or age-related
I want to
effects?
ask you about mitochondria and aging. So we have reactive oxygen species. We have peroxide ion, superoxide anion, hydroxyl radicals, hydrogen peroxide. Why does the respiratory chain produce reactive oxygen species, and what is the respiratory chain?
All right, let's dive into mitochondria, aging, and reactive oxygen species, ROS, keeping it concise. The respiratory chain, aka the electron transport chain, ETC, is a series of protein complexes, 5R, in the mitochondrial inner membrane that transfers electrons from NADH and FADH2 to oxygen, pumping protons to drive ATP synthase for ATP production. During this, some electrons leak, mainly from complexes 1 and 3, and react with oxygen to form ROS like superoxide anion, hydroxyl radicals, hydrogen peroxide, and peroxide ions. These ROS are highly reactive, damaging mtDNA, proteins, and lipids. The ETC produces ROS as a byproduct because electron transfer isn't 100% efficient, especially under high energy demand or stress. In aging, accumulated ROS damage impairs ETC function, reducing ATP and increasing mtDNA mutations, which worsen oxidative phosphorylation decline. Clear? Want more?
So what is the difference between the electron transport chain and the respiratory chain?
There's no difference. The electron transport chain, ETC, and respiratory chain are the same thing, just two names for the mitochondrial process. Located in the inner mitochondrial membrane, it's a series of protein complexes, I4, that transfer electrons.
Okay. So what does homoplasmy and heteroplasmy mean in regard to mitochondria and genetic bottleneck?
Homoplasmy and heteroplasmy describe the state of mitochondrial DNA, mtDNA, within a cell or individual, tied to mitochondria's maternal inheritance and the genetic bottleneck. Homoplasmy means all mtDNA copies, hundreds to thousands per cell, are identical, either all normal or all carrying the same mutation. Heteroplasmy means a mix of normal and mutant mtDNA copies coexist in a cell. The genetic bottleneck occurs during oocyte development in the female germline, where only a small subset of the mother's mtDNA is passed to the egg, amplifying or reducing specific mtDNA variants in the offspring. This bottleneck can shift heteroplasmy levels, affecting whether a mutation dominates, potentially causing disease, or stays minor,
I don't
impacting.
understand. I don't understand. Tell me again.
No problem. Let's simplify homoplasmy, heteroplasmy, and the genetic bottleneck for mtDNA. Homoplasmy is when all mitochondrial DNA copies in a cell are the same, either all normal or all with the same mutation. Heteroplasmy is when a cell has a mix of normal and mutant mtDNA copies. The genetic bottleneck happens in the mother's egg development, where only a small number of her mitochondria and their mtDNA are passed to the egg, randomly amplifying or reducing mutant mtDNA. This can make the offspring cells mostly homoplasmic, same mtDNA, or heteroplasmic, mixed, affecting health or aging gladly inherited diseases and aging. Clear? Want to focus on examples or implications for aging?
So homoplasmy means it's either completely normal or completely abnormal. And what is genetic bottleneck?
Exactly. Homoplasmy means all mitochondrial DNA, mtDNA copies in a cell are either completely normal or all carry the same mutation. Heteroplasmy is a mix of normal and mutant mtDNA. The genetic bottleneck is when during egg development in the mother, only a tiny fraction of her mitochondria with their mtDNA are passed to the egg, not all of them. This random sampling can boost or dilute mutant mtDNA in the offspring.
So genetic bottleneck is there's going to be random. Why? How does the name relate to the situation? And what's the threshold effect?
The term genetic bottleneck describes how only a small, random subset of a mother's mitochondrial DNA, mtDNA, is passed to her egg during oocyte development, like squeezing a large population through a narrow neck, limiting which mtDNA variants reach the next generation. This random sampling can amplify or reduce mutant mtDNA, leading to homoplasmy, all normal or all mutant, or heteroplasmy, mix of both. The threshold
It's
effect.
like we have 10 people, one is angry, one is happy, one is sad, one is evil, one is virtuous. And basically what's going to happen is we're only going to choose 2 of them. So our new population might be a bunch of happy people or a bunch of virtuous people or a bunch of whatever.
Exactly. Your analogy nails it. The genetic bottleneck is like picking just a couple of those 10 people, say 2 at random, to start a new group. If you happen to pick the happy and virtuous ones, your new population might lean heavily that way. Or if you get the angry and evil ones, it shifts differently.
Okay, let me ask you a bit about myopathies and tell us what myopathies mean. So mitochondrial myopathy specifically. So we have, let's say, a mutation in mitochondrial genes in the mitochondrial DNA. Now we have a reduced capacity to produce ATP. What are some tissues involved, like neurons, myocytes, skeletal muscle cells, beta cells of the pancreas, and how are they now less able to tolerate this? Well, I understand how they're less able to tolerate the ATP production, but maybe my question should be instead, how does ATP why is ATP suddenly affected with this mutation? Give us an example of a mutation that will affect it. Maybe a neuromuscular disease or something.
All right, let's dive into mitochondrial myopathies, keeping it concise. Myopathies are diseases of muscle tissue causing weakness or dysfunction. Mitochondrial myopathies specifically result from mutations in mitochondrial DNA, mtDNA, or nuclear DNA, affecting mitochondrial function, reducing ATP production via oxidative phosphorylation. A common mtDNA mutation, like the M3243A mutation in the MTTL1 gene encoding a tRNA, disrupts protein synthesis for electron transport chain ETC complexes I, 3, 4 or ATP synthase, lowering ATP output. Tissues with high energy demands, skeletal muscle cells, myocytes, neurons, cardiac muscle, and beta cells of the pancreas are hit hardest.
Okay. So, okay.